Hello and welcome back to the Biochemistry for Health Sciences channel. Today we'll talk a little bit about these tests, these chemicals called AGEs or advanced glycation end products, hemoglobin A1c, which is a commonly ordered lab test for diabetes, as well as fructosamine, another lab test that is useful for diabetes. So in the previous videos, we saw how aldehydes are very reactive in water, and they can react with amines. And the carbohydrate can then get attached to the protein. And since proteins do most of the things for cells, as well as provide structure, the cell structure and function can be affected. So let's look at this a little bit more closely. So one of the toxic products that we get when aldehydes or ketones react with proteins or other amines or amines present in other biomolecules. So this reaction between aldehyde and amine. And you can see here an example, glucose reacting with some amine substance, whether it's a protein or lipid or whatever. The aldehyde amines react to give you what we call a Schiff base. This is what organic chemists talk about. This Schiff base is very unstable. So most of it usually breaks back to give you this. But some of it can be converted to this product that we call an amidori product. Okay, an amidori product. This is stable, the amidoric product. And this product can then break down to give you other aldehydes that can go on to react with other proteins and give you what we call advanced glycation end products. Similarly, the Schiff base, you can see, can give you products, aldehydes that can react with other proteins or biomolecules to give you AGE. Glucose itself can give you other aldehydes and AGEs. And through glycolysis, we have these 3 sugar intermediates. You can see the aldehyde. Here's a ketone. Okay, and again, these 3 sugar intermediates cannot form cyclic structures. They are very, very reactive. So let me erase this. So you can see these kinds of structures. They're very, very reactive because they cannot form cyclic structures. You see, very reactive aldehydes and ketones. Okay, and they all can react with proteins and give you advanced glycation end products. And how do we know chemicals like this are very dangerous? Because they are genetic diseases where the infants cannot metabolize these 3 carbon sugars, and they start accumulating in the cells. And when they accumulate, we see a lot of AGEs, and the infants basically die very young. So fortunately, this reaction between aldehyde and amine is a slow reaction. However, when your blood glucose concentration is very high for a prolonged period, so the higher the concentration of the glucose, the faster the reaction is going to be. Okay, so glucose will react with any proteins, but the problems happen especially with long-lived proteins that are around for a long time, because a lot of proteins, when they're made, they don't live around too long. So even if they're glycosylated, they are destroyed. So the chances of them being reacting with glucose is very low. But the long-lived proteins such as hemoglobin, which is found in red blood cells, your elastin, collagen, and the proteins in the islets, they live around for a long time, and they are more susceptible to becoming glycosylated, reacting with carbohydrate like glucose, and then forming advanced glycation end products. So why are these AGEs dangerous? Because AGEs actually trigger inflammation. They are actually receptors for AGEs. We call it RAGEs. Okay, receptors for advanced glycation end products. And these receptors are found on endothelial cells in your blood vessels, your smooth muscle cells, immune cells, and other cells. So And one of the things that ages do, these products, really, they do protein cross-linking. They link two proteins together, okay, covalently. And when that happens in blood vessels, the like elastin, the elasticity of the blood vessels is affected. And it also decreases the stickiness between cells. So the endothelial cells, you can see their stickiness is reduced. These cross-linked proteins, the cross-linked proteins then also start trapping particles such as LDL and albumin. And when LDL gets attracted to the blood vessels, the endothelial cells, that eventually leads to arteriosclerosis. And when albumin starts sticking or starts getting trapped, on these cross-linked proteins between the endothelial cells, then we start seeing more and more proteins getting deposited on the endothelial cells. And that happens, for example, in the kidneys in diabetic patients. It's very difficult to degrade the proteins that are cross-linked or the proteins that are glycated. So these proteins eventually start accumulating and start giving you blood vessels that are kind of abnormal, very thick but abnormal. So this reaction here, glucose reacting with hemoglobin, if this was hemoglobin, to give you the amidary product, this is the basis for your glycosylated hemoglobin test. Since hemoglobin stays in your red blood cell for 120 days, so basically this glycosylated hemoglobin test gives you an idea of your average blood glucose for 120 days. The major hemoglobin in adult is HbA. About 6% of HbA, hemoglobin A, is A1. Okay, and A1 is further divided into 3 types. Okay, and we can use techniques such as chromatography to separate these various types. There's A1A, A1B, and A1C. A1C is the major form of this A1. So A1C is actually the hemoglobin attached to glucose in your red blood cells. Okay, so A1C usually, if it's more than 6.5% on 2 separate tests, that's a strong indication of diabetes. So it's one of the best tests for diabetes today. A1A is composed of actually 2 fractions. One is hemoglobin, which has reacted with a sugar fructose 1,6 bisphosphate, and the other is hemoglobin, which is reacted with a sugar glucose 6-phosphate. Okay, so that we don't usually report that part typically in lab tests. A1B is basically hemoglobin attached to pyruvate. So A1C is what most labs measure, A1C. Okay, and that's the one where the glucose has reacted with hemoglobin, glucose and hemoglobin. There is a formula available in literature which relates your mean blood glucose to the A1C. So it's 33 times A1C minus 86. So a patient with 16 A1C, you just put 16 here. So that's 33 times 16, whatever the number is, minus 86. And that comes out to be 445. So a person with an A1C of around 16 really has a glucose level of, an average glucose level of 445 milligrams per deciliter for the last 120 days. Okay, remember the normal glucose level is around 100 or less. So that's a pretty high level. Besides the A1C, there is another test available called glycated albumin, or the fructosamine test. In this case, the protein here is not hemoglobin, it is the protein albumin. Okay, so obviously glucose can react with any protein. So albumin has a lifespan of 20 days. So glycated albumin is albumin that has glucose attached to it. Albumin is very important in the body. We'll talk more about this in metabolism. It binds all kinds of chemicals, including fatty acids, and various hormones and so on. And it's also a very, very good antioxidant. So once it is glycated, the activity of the protein is affected, the ligand binding as well as the antioxidant activity. So fructosamine gives you an idea for the mean blood glucose for about 20 days. That's the lifespan of the albumin. This is a good test for people who have abnormal hemoglobins. Okay, the patients with abnormal hemoglobins, different type of hemoglobins, the A1c test is not very good. It's not very reliable. So physicians can always order the fructosamine test. Reference range around 200 to 300 micromoles per liter. Diabetes is more than, I think it's more than 390. It's a mistake here. It's more than about 380, 390 micromoles per liter. So before we leave this discussion on AGs, I would like to mention another definition that is especially important in pathology. Okay, these words are used quite often in articles, medical articles, and that's PAS positive. What do you mean by saying PAS positive? The tissue was PASF positive or had strong PAS reaction. PAS stands for periodic acid Schiff reaction. So let's look at this example here. Here's a normal glomerular capillary. Here's one from the diabetic patient. Prolonged glucose levels are very high for a long time, so uncontrolled diabetes. And you can see this increase of all the AGEs cause increased deposition. So there's a lot of, the basement membrane is very rich in all kinds of deposited material, cross-linking and so on. Now, if we were to stain this with a stain called PAS, periodic acid, PA is periodic acid. Periodic acid basically takes the sugar. So remember, there's a lot of sugars attached here to the proteins. So a lot of sugars attached to the proteins compared to a normal. Normal, there's very little sugar attached to a protein, but here there's a lot of sugar attached to the protein. So the sugars, when it reacts with periodic acid, these sugar OH become aldehyde. And then the aldehyde will react with these amines on the stain called the Schiff's reagent. And when these two react, it gives you a pinkish color. So it gives you a pinkish color. So when you look at under a microscope, at the glomerular capillaries of a diabetic patient, you see this very, very strong pink. This is your PAS, your strong PAS reaction. Compared to this here, you hardly see, in a normal person, you hardly see much of the PAS positive reaction. Okay, so periodic acid Schiff reaction. This is a stain to look at the carbohydrate part, the carbohydrate that's attached to the proteins that have been deposited in these capillaries. Okay, so that's all for this video. I understand that Kohli is traveling. This is the last picture we got from his travels. And he promised to send more pictures for the next video. So this is me, Professor, telling you goodbye and hoping we will see you in the next video. Until then, adios.